Hi there, my name is Brittany Size, and I will be presenting to you my journal article presentation for cellular biology. Today is September 20th, 2019, and this is for Professor Joan Hudson's class. So my protein given was Rubisco, and otherwise known as ribulose biphosphate carboxylase oxygenase. And this is an enzyme that is presented in chloroplast, involved in fixing its atmospheric carbon dioxide during photosynthesis and in oxygenation of the resulting compound during photorespiration. This is also found as part of the Calvin cycle. So my journal article right here is going to be on the stability and activity of Rubisco and chestnut plantlets transferred to ex vitro conditions under elevated carbon dioxide. This article was written in July 2005 and is going to be by Luisa Carvajalo, Maria G. Squivel, and Sarah Amancio. So in abstract, what experiment was done was seeds of the chestnut plantlets were taken from the organism in ex vitro to test tubes in vitro, and differences in light dimness and trying to stress out the Rubisco to see how much carbon dioxide would be produced after, and see how the plants grew differently based on the light and the environment given, the light intensity and the gas exchange being limited in an airtight container. So a couple things I found very interesting about this article was they showed it against other woody species and how that differentiated. They were saying that usually the carbon dioxide doubles concentration enough to increase the photosynthetic rate of woody species. It didn't do everything that they really thought it was going to do. It shows over here that when growing at elevated carbon dioxide, plants performing C3 photosynthesis show short-term gains of photosynthesis rates and long-term downregulation of photosynthesis or acclimation to elevated CO2. In our systems, lower Rubisco activities were measured under HCO2, mainly in association with 8th L. Acclimation is generally accompanied by a substantial increase in soluble sugars and starch, and by decline in Rubisco content, activity, and activation state. So here's my journal article, and I'll see you guys next week. Thank you.